I am pleased to be able to tell you a story start to finish. So much of what happens is chunked up into each of its papers, but I wanted to take this time and tell a story from start to finish, and as the title indicates, it's potentially a very interesting and relevant story. Our group, Dr. Waxman, has based a lot of the work on the idea that studying rare diseases is a window into understanding basic physiological and pathophysiological processes. And as Martin indicated, today's story will focus on NaV 1.7, and there are 9 members of the sodium channels expressed in humans, and in pain, in the dorsal root ganglia, 3 are highly expressed, 1.7, 1.8, 1.9. The 1.8 and 1.9 may be talked about by other speakers over the course of the next 2 days, but today I will be focusing on the 1.7. And the disease that we're focusing on, the story I'm telling, is primary inherited erythromelalgia. And the inherited part allowed it to be determined that NaV 1.7 is the gene underlying these patients' severe pain syndrome. And this is art drawn by the patient themselves to try to convey what kind of pain that they exhibit. And clinically, we talk about burning pain in the extremities, the hands and feet, and they turn red. And for most of the patients, there is no help. They try one thing, another thing, and nothing seems to help. So these patients are in a very strong need to find therapies that can help them. So finding therapies for pain is obviously a big question. And so in America, we're suffering from adverse outcomes from pain drugs. And so if we could work towards understanding neuropathic pain, for which IEM is a model, maybe we could develop next generation painkillers that would help not only our patients, but everybody in general. And this whole idea of making the NaV 1.7 a primary target is boosted by the finding that the same gene, when mutations that cause loss of function are found, result in a phenotype of congenital inability to experience pain. So here are rare examples of patients who do not have functional 1.7, and yet they're conscious, they're relatively normal. So except for they're having to monitor their environment to see if they've done something to hurt themselves, they're relatively normal. So this natural experiment has suggested that if drug companies could target 1.7 and inhibit it specifically, that it should be able to reduce pain without causing serious side effects. It's as good a setup as you could ask for a targeted drug campaign. And indeed, numerous large pharmaceutical companies have initiated campaigns to develop drugs to targeting 1.7, and hopefully will result in safe and effective new pain compounds that will have none of the side effects like the opioids do. And some of the people in the room may have touched some of these compounds, but for the patients that are experiencing IEM, drug development takes many, many years. And so even though they're given some hope that eventually a compound will help them, we wondered, is there something we can do to help our patients more quickly? And so one of the things we can do is if by chance a drug-responsive patient population is found, we can try to understand why and perhaps give us some clues on how to expand that to others. And there is a family that reported some relief when they were put on carbamazepine. It's an already approved drug commonly used for epilepsy or other hyper-excitability things. And our group, Dr. Waxman's group, were very good at studying the biophysical properties of ion channels, and we put this variant into a heterologous expression system, and we tried to understand how does this channel work. And for, in voltage clamp, we put on a series of depolarizations, measure the current that gets elicited, and can derive a curve that characterizes the activation, what amount of stimulus will cause the channel to open. And when you compare the mutant to the wild type, which is this barely visible gray one, it's shifted to the left, which means that these channels are more easily opened by depolarizations. And this is the common biophysical change that has been seen in a number of IEM patient family mutations. And as we studied this particular mutant, we saw in the clinical workup that it was carbamazepine sensitive. And so we tried to determine what is going on with carbamazepine. We were not able to detect any change in the IC50s for carbamazepine or the use dependence. But we did see that if we pre-treated the cells with carbamazepine and then redid the characterization, it's this sort of novel shift of the activation curve back towards wild-type values. So it's almost like the drug is a chaperone and helps the perhaps slightly misfolded channel fold more like wild-type and normalizes the properties of the channel. But to better understand that, we wanted to improve our way of thinking about it. And so we expanded our structural biology capabilities in the lab. And what we're seeing here is a homology model of a human NAV 1.7 channel based on reported bacterial sodium channel structures that have been released. And so we're moving from this linear topology model where you can look at the site of various mutations and maybe get an idea to realizing that the residue at position 400, which is normally a dysvaline, was pointing directly toward a residue which is normally serine 241. And we happen to know that there's another family that suffers from IEM when the serine is mutated to threonine. So the S241T mutation is also an IEM mutation. And we thought this was very interesting. Is there a reason that maybe this region of the molecule is very critical in controlling the gating, the control of the voltage? And so we wanted to test this. I mean, this is a homology model. We don't actually know this is true in human, but we can test this. And one of the ways we tested it was to do multi-state modeling, which is to move the channel from closed to open to inactivated and see how all the parts are moving together. And we found that the two residues moved together in all of our modeling. But then even more convincing to us was to do thermodynamic mutant cycle analysis. And the idea here is if an amino acid is sharing the same energetic pathway, then if you mutate, you make the V400 mutation, you have a shifted. If you make the S241T, you have a shifted. But if you make both and they share the same pathway, then you do not get any additional shift, which is what we saw here. So that suggests that these two residues are energetically coupled. And of course, we can do the same mutant cycle analysis with a IEM mutation, which is in a different domain in the S6, sort of analogous position as the V400. And in this case, each residue by itself has its shift, but when we make the double mutation, we basically get addition. So in this case, these two residues are acting in parallel, not in the same. But it bolsters the case that the S241T and the V400 are interacting, that they're part of the same energetics. So the homology model is perhaps telling us something real about the structure. So the two amino acids are shared, and we further asked, since the V400 is responsive to carbamazepine, is the S241T responsive to carbamazepine? And indeed, this unusual shift that we already characterized for V400 also occurs for the S241T. So we're thinking this could be, we've based on entirely lab-based in vitro assays, we could have predicted that we found a drug that will pair for a particular mutation. But before we rush into, we wanted to further test that carbamazepine is the right thing to do. And so in addition to our expertise in voltage clamp biophysical characterization, we've been developing sort of pain in a dish assays. And what we're doing here is introducing our mutants into rodent DRGs. So we'll have a rodent DRG, and we introduce either the mutant human 17 or the wild type human 17, and we'll do current clamp recordings from these populations, average them all together, and see whether we actually are altering excitability. And among the excitability measures is threshold. So we inject very finely graded current injections until we get the all or nothing action potential. And indeed, this shows that our DRG model will fire a threshold at low few hundred picoamps. And if we're expressing this mutant, the threshold is reduced, consistent with what we thought based on the biophysics. But if we pretreat with the carbamazepine, the same cells are now elevated in their threshold back towards wild type values. So again, the way we predicted in biophysics is turning out in our pain in a dish assay. Not only do we look at threshold, but we can look at super threshold and ask for a given super threshold, how many spikes do we get? And again, in the presence of carbamazepine, the S241T is abrogated in the number of spikes. And when we plot it as a number of current injections, we can see a clear reduction in the excitability of these neurons. So based on all these levels of evidence, we decided, well, first we had a discussion. Should we just go tell the physician, you can try carbamazepine and we think it'll work? But on the other hand, if we perform a clinical study, then we can publish it, and then we can perhaps help additional families that might share the same mutation site, and it would spur further development of maybe perhaps some additional pairings of drugs with results. So our patients have been very supportive of our group, and so they were practically begging us to participate in studies. And so one of the first studies we did was just to characterize what kind of pain that they exhibit and how its kinetics are. And each patient is different. So some people have very fluctuating pain during an attack, and others are more sustained. And so these kind of measures increase the the complexity of trying to design a study, but we decided to go forwards with a study that does both self-journaling of their pain, their reported pain score, but we decided to also add functional MRI imaging. And so we did a crossover study. It's only 2 patients, so of course, we can't make big claims about statistical significance. But even though self-reported pain diaries may be prone to the placebo effect, the patients want to please the researchers and say, but it's going to be much harder for a patient to fool brain scans. So before, during the 2 arms, they're visiting and undergoing fMRI scans. And I'm pleased to report that like the original V400 family, it's not that that carbamazepine is capable of blocking all pain, but these indicate that the number of attacks and the duration of attacks are shortened by being on the drug. And in addition, the fMRI, which was performed by a physician, Paul Geha, he helps us understand that there are areas of the brain that light up, that he can determine are related to the pain. And when they're on vehicle, certain areas light up that are more typical of neuropathic pain signals. And then in the presence of carbamazepine, different areas of the brain are lighting up, more consistent with a shift from a chronic pain state to an acute pain state. So we both got a positive response from the patients themselves who report that they're able to sleep at night, that they don't have as many attacks, and with this hard-to-fool assay, clearly the carbamazepine is doing something in the experience of these patients while they're on the drug. So it's a story that's start to end. From the first reports identifying 1.7 as being underlying EM pain, the CIP, so drug development is starting somewhere, targeted drug development, targeting 1.7 is starting somewhere. We characterized V400 around 2009, the S241 by 2012, and at that point we got the design, the IRB approval, and the actual performance of a trial in just 2 to 3 years, which is pretty impressive, I think, to help patients that carry this mutation. And so it's sort of a model that we can move forwards and perhaps help other patients until waiting for the more broad spectrum, help everybody kind of drugs to come along. And things are in the works. We have definitely identified other mutations that are energetically coupled and so are carbamazepine sensitive. But I think just in the same algorithm, if we scan the literature and we find a patient that maybe responds to some other drug and look at the drug interacting with that particular location, we might be able to not only help, allow that patient to continue to be helped, but then identify the other mutations that might be selective to that drug. So we're thinking about how to pair drugs to patients, which is precision medicine, which is really cool to me. But how can we speed this up? We're very good at doing manual patch clamp. So we're investing in auto ePhys instruments so that we can do the voltage clamp experiments more quickly. And in our pain in a dish model, the current clamp experiments are arduous. They take time too, but we're investing in multi-electrode arrays. And we're also trying to improve our pain in a dish assays by moving towards patient-derived iPSC derived sensory neurons. This is our MEA instrument. It's plate-based. Each well of a plate will have an array of electrodes. If a neuron happens to land on or near the recording electrode, it will be able to detect the firing of these cells. And this is animation where it's a heat map, so the 8x8 array of electrodes are analyzed, and the firing rate is illustrated. And so for our S241T, it's highly active and temperature sensitive and drug responsive. So we have a higher throughput assay. And then in the same assay, you can see that S241T derived sensory-like neurons are firing much more than sensory neurons derived from healthy donors, and drug is effective. So we're looking forwards to seeing if we can advance precision medicine for our patients. And hopefully we'll extinguish the pain. Thank you. Oh, and we're a big group. I get to tell the story. I contributed to much of what I told, but there are many, many people in our group who participate. All right, thank you.
Thanks, Mark, for a great talk of bench to bedside in action. Questions?
I guess the one problem with understanding the effects of carbamazepine is that it's not a very specific sodium channel blocker. So do you have any idea where the site of action is in these patients? So the fMRI rather suggests there are central actions as well as...
There may very well be. There's some debate or discussion about whether an extremely specific blocker for 1.7 will be as achievable, and that you can achieve clinical efficacy with a slightly dirtier compound. So we have a compound that seems to do something specific to these mutations, but maybe what it's doing to the mutation plus what it's doing everywhere else is part of its efficacy for these patients.
I was just curious, of all the anticonvulsants, why did you choose carbamazepine?
Well, it was based on the clinical write-up of the V400 patients where they tried 2 or 3 different things, but carbamazepine worked for them. So that was the primary thing. I will say that we've tried some analogs of carbamazepine, and they're not doing the same thing to our variants. So even though they're doing what they should for use dependence or shifting the availability curves, they are not doing this sort of unique normalization thing. So something is specific about carbamazepine on that signature.
Is there any information about patients suffering from neuropathic pain, whether they also have a shift in the activation of NaV 1.7? Because that might mean they're more responsive to compounds like carbamazepine then.
So our pain in a dish model works extremely well for these strong mutants. Some of the things we've attempted is to put on inflammatory soup, and we do get hyper-excitability, but with so many channels already in the neurons, it's not as easy to specify that it's doing something just to 1.7.